It's a very nice summary of the exciting combinations. So with the 3 novel oral drugs that are currently approved on the market for CLL, ibrutinib, adalimumab, and venetoclax, so the question is, how do you best use them? How do you combine them? You can combine them with monoclonal antibodies. You can combine them with other class of novel oral therapies. And there are also studies that are combining them with conventional immunochemotherapy. And then the next question is, what's next, right? What's coming? I think that's probably one of the most exciting things to look at. This is a very busy slide. You don't have to read it, but it's just really it came from a review article published in Nature Review by Dr. Berger and Dr. O'Brien. It really shows you that there are a lot of things we have learned over the last several decades, both in terms of understanding the biology and prognostic factors for CLL, but also the evolution of the different treatments. And you can see that in the recent evolutions is the BTK inhibitors, the PI3 kinase delta inhibitors, the BCL2 inhibitors, and then what's coming next are probably second generations and also the immunotherapies. So I would like to kind of give an overview of what I think are the most exciting things that are in the pipeline that are coming probably, I'm hoping, in the next several years to become available for CLL. So these are all things that are being actively studied. I divide them into 2 big groups. One is novel oral pathway inhibitors. The other is novel immunotherapies. So among the novel oral pathway inhibitors, we talked about the B-cell receptor pathway inhibitors. So that includes the BTK inhibitors that we already know about, but now there are second generations of BTK inhibitors. There are PI3 kinase inhibitors such as adalimumab, the first generation. Now there are several other PI3 kinase inhibitors that are being studied. There's also the Syk enzyme. So all of those enzymes are important molecules along the B-cell receptor signaling pathway, and they all have very similar treatment effect and unique mechanism of action. The other class is the BCL2 inhibitor, which you already heard from Dr. Venugopal with the venetoclax data, which is very exciting. And then there are some other pathway inhibitors that we'll touch on. Among the novel immunotherapies, we talked about novel monoclonal antibodies, which we're all very familiar with. We've been using monoclonal antibody-mediated immunotherapy for the last 2 decades already. And there's some new ones that are more glyco-engineered. There are also bispecific antibodies that are trying to conjugate T cells and the B cell targets. There are the immunomodulatory agents, which you're very familiar with in the myeloma data, but they're also being studied in lymphomas and CLLs. And then there are some data on immunocheckpoint inhibitors, which I won't go into much details, but we'll touch just briefly on. And then I think probably one of the most exciting things that's coming up is the CAR-T or CAR-NK therapy. So looking at CLL, the malignant B cells are not living alone. They're not really growing or expanding on their own. They heavily rely on the interaction or crosstalk between the malignant B cells and the microenvironment they're living in. So there are stromal cells in the lymph nodes, in the bone marrow, and even nursing cells in the peripheral blood that are sending these important signals to promote B cell proliferation and survival as well. So the B cell receptor is one of the most important receptors that transmit those kind of signals from the microenvironment. So there are, as we talked about, there are several enzymes you can target, include the BTK, the PI3 kinase, and the Syk enzyme, which are all sitting in this pathway. And then you can also target B cells by targeting surface antigens, such as the CD20 antigen and the CD20 monoclonal antibody has used for a very long time. And then you can think about other immunotherapy using the T cell cellular immunotherapies. So this is the B cell receptor signaling pathway, and you can see there are a number of tyrosine kinases sitting in this cascade, BTK, Syk, and PI3 kinase. And often cells that are developing resistance to ibrutinib treatments have mutations in BTK enzyme or PLC gamma 2. Those were the known mutations that could potentially cause resistance. So for B-cell receptor pathway inhibitors, the three classes, BTK inhibitors, the first generation is the prototypic ibrutinib. And then the second generation inhibitors are primarily developed in order to have more specific inhibition of BTK. Because we know that ibrutinib not only targets BTK, the proton tyrosine kinase, it also has several off-target activities for example, EGF receptor, for the TEC enzyme, for the ITK enzyme. So we believe that a lot of those ibrutinib-related adverse events were probably mediated by those off-target tyrosine kinase inhibition. So the second generation BTK inhibitors were designed to be much more specific for the BTK enzyme alone and have much less activity against the other enzymes, and therefore in hope for less potential adverse events. Similarly, second generation PI3 kinase inhibitors are also designed to be more specifically targeting the PI3 kinase delta, but also chemically structured to try to minimize the effect on potential toxicities. So all of the B-cell receptor pathway inhibitors seem to have very similar way of acting. Because of their interruption of the crosstalk between the malignant B cells and the microenvironment, it's altering the B-cell trafficking. So it's really having pretty interesting response early on. So you can see that before therapy, patients often have lymphadenopathy, hepatosplenomegaly. If you look at their peripheral blood over time, before the treatment, they're having increasing lymphocytosis, and meanwhile, they're having gradually declining of their bone marrow function, including the hemoglobin and platelets. So that's before the treatment. Once patient starts with the B-cell receptor signal pathway inhibitors, such as ibrutinib or idelalisib, then what you can see is that the cells actually rapidly are leaving those compartments. So the lymph nodes, as a result, are very rapidly shrinking, and the spleen and liver are also quite rapidly improving. Usually, we can see some results within the first couple of weeks. And meanwhile, where did all those cells go? They went out to the peripheral blood. So you can often see this peak in the lymphocytosis. So that's called treatment-related lymphocytosis. They often peak around 4 weeks or so, and then will gradually calm down. And meanwhile, you can see that the bone marrow function is improving, so with improved hemoglobin and improved platelet counts as this is happening. So this is a quite unique characteristic response pattern for the B-cell pathway inhibitors. So I talked about that the second generation inhibitors are designed to be more specifically targeting BTK and try not to target some of the off-target enzymes. Among them, acalabrutinib, or ACP-196, is already FDA approved for mantle cell lymphoma, and it has a lot of data on CLL as well. The other one is the BGB3-111, or zanubrutinib. There are a lot of brutinib you have to pronounce. So there are a lot of ongoing, really interesting clinical trials using those agents. And then the other one, there are 2 other BTK inhibitors by other manufacturers. So the top couple of them were all irreversible BTK inhibitor. The last one, the SNS062, is a reversible BTK inhibitor. And the design for that is in hope that it may be able to overcome or prevent the development of the resistance to BTK inhibition. So I'm not going to have time to go over all of the different molecules in details, but just to give you one example of the second generation BTK inhibitor, acalabrutinib. So this is a phase 1/2 study of acalabrutinib in relapsed refractory CLL. You can see that the response rate is also quite significant, similar to ibrutinib. That you can see most of the patients actually have pretty significant response. This is at different dose levels. And then the 100 milligram BID dose is the final recommended phase 2 dose. So with this, the overall response rate is over 90%, and there's about 13% of complete response. And then similar to ibrutinib, you can see that the treatment response really improved over time. So because this is a continuous oral treatment, very different from the limited duration treatment like immunochemotherapy, where you give a couple months of treatment and the best response would be once you finish all those treatment. Because of the continuous nature of the treatment, the response often improve over time. So you can see that at the 6-month mark, about 50-some% overall partial response rate. But if you look over at 1 year, it improved to 70%, and then at about 2-year mark, almost 100% response rate. Okay, so definitely be patient, and you can definitely see the quality of response improve. There is a response category called partial response with lymphocytosis, which is not counted towards the conventional partial response. But those are the patients who have significant lymph node and bone marrow response, but they are having those treatment-related lymphocytosis. So now in the new guidelines, when you're looking at the novel therapies, these should be included, counted as total response, overall response. So it's just this is a busy table, but just to show you that different BTK inhibitors, how they compare to that ibrutinib. And so basically overall response rate are all in the 80-90% rate. And complete response is not very common usually in the single digits. The progression-free survival, because of the newer agents have shorter follow-up time, but it all seem to be fairly similar, at least comparable efficacy-wise. Side effect profile, I think there still need to be more longer-term follow-up. But for example, there are some unique side effects such as headache with the acalabrutinib. The thought is that because it's probably crossing the blood-brain barrier better compared to the ibrutinib. In terms of what we really want to see is there any improvement in the atrial fibrillation rate? Is there any improvement in the bleeding risk? So that I think there's no direct comparison yet. There may be some anecdotal suggestion that it looks promising. However, there's no strong evidence of that yet. So there are some actually side-by-side phase 3 study to compare different BTK inhibitors ongoing. So we hopefully can learn more about the side effect profile from those. So that's the second generation BTK inhibitor. Now we're going to talk about PI3 kinase delta inhibitor. So the prototypic one is the first PI3 kinase delta inhibitor that's approved, adalimumab. So looking at PI3 kinase, there are 4 different subgroups, the alpha, beta, gamma, and delta. The gamma and delta are more specifically expressed in the leukocytes, and they're important for the white blood cell biology. So alpha, beta are more in the metabolism. So if you have a pan-PI3 kinase inhibitor, there are going to be a lot of other potential metabolic side effects. So that's why the more specific gamma and delta inhibition is more suitable for use in lymphoma treatment. So this is the first, the phase 3 study that established adalimumab as a treatment for CLL. So this is a study comparing adalimumab plus rituximab versus placebo plus rituximab in relapsed refractory CLL. You can see that this is the waterfall plot looking at the response of lymph nodes. So there's significant reduction in the lymph nodes for the adalimumab plus rituximab arm. Virtually all of the patients had improvement. Versus the rituximab arm, much more moderate. And then this has shown that both in terms of the progression-free survival and overall survival, the addition of adalimumab to rituximab seems to have been superior to placebo. So the problem with adalimumab is that there are some potentially concerning side effects. In particular, besides the common ones, in particular is some autoimmune phenomena. For example, autoimmune hepatitis. So transaminitis is fairly common for adalimumab treatment. That tends to happen more in the first couple months. And then there are other autoimmune, especially the immune-mediated colitis. That can happen anytime during the treatment course. And often if patient develop immune colitis, then they have much more significant diarrhea that may have to lead to treatment interruption or even discontinuation. So the second generation or other PI3 kinase inhibitors were trying to improve upon those toxicity profile. There are the dual PI3 kinase inhibitor, duvelisib, which structurally is related to adalimumab, but is not only inhibiting delta, but it's also inhibiting the gamma isoform of the PI3 kinase. The side effect profile is, I think, maybe moderately improved, but not significant. There are other second generation PI3 kinase delta inhibitors. I think the one that's very interesting is the umbralisib, which we're going to look into more details. So here is umbralisib comparing to adalimumab versus duvelisib. So these two are PI3 kinase delta inhibitor. This one is both delta and gamma dual inhibitors. You can see that chemical structure of the umbralisib is very different from that of these two. And I think as a result, that may have explained some of the different toxicity profiles that you're seeing with umbralisib. And also, if you look at the selectivity among the different isoforms of PI3 kinase, the umbralisib seem to have more selection, so much less targeting the alpha and beta, and then it's in terms of the activity for delta, they are all very similar. So umbralisib had been shown to seem to have improved, based on the early clinical trial data, there seem to have improved profiles in terms of liver toxicity and the autoimmune colitis. So that's very encouraging. And it's also something that's a longer half-life that allows once daily dosing. And it's interestingly, it targets another enzyme called the casein kinase 1, epsilon. So this is a protein that may actually regulate the regulatory T cells, so Tregs, and that's their thoughts. That's why they think there may be a better autoimmune toxicity profile because of the Treg regulation. So here's just a comparison of different PI3 kinase delta or dual PI3 kinase inhibitors in terms of the response rate. And then in terms of the progression-free survival and the overall survival. So they're all quite similar. So they're all very effective inhibitors of the PI3 kinase and therefore the BCR signaling pathway. I think the more interesting thing to look at is the adverse events profile because they're efficacy-wise, they're all quite effective, but I think we're really trying to improve upon the toxicity, especially the concerning toxicity such as the autoimmune hepatitis, colitis, or pneumonitis associated with adalimumab. So, all right. So venetoclax, you already heard a lot from Dr. Venugopal about venetoclax. This is a BCL2 inhibitor that really targets directly the apoptosis machinery. So this is a whole new family of molecular targeting therapy that's different from the previously discussed BCR pathway inhibitors. So BCL2 is an anti-apoptotic protein that is protective for the cells that's actually blocking the apoptotic pathway. So by inhibiting the BCL2, you're actually releasing the cells and allowing them to progress normally into the apoptosis, and the cells can actually very rapidly undergo apoptotic cell death. Just to show you how effective it is, this is just a simple example of looking at the absolute lymphocyte count, how quickly it's responding to a single dose of venetoclax oral therapy. So you can see that within 8 hours, there's a dramatic decrease in the peripheral lymphocyte count. And meanwhile, you can actually see an increase in the LDH, which is an indication of tumor lysis. So the reason venetoclax, one of the main things to watch out for in terms of adverse events is really because how effective it is. It's just a very rapid onset of triggering the apoptosis, and therefore the tumor lysis risk is something you really have to watch out for. So here's just a study that you probably already heard most about in terms of venetoclax single agent versus combination therapies. So both in terms of combining with anti-CD20 antibody and combining with ibrutinib, it seemed to be a good partner to combine with the other class of treatments to give you deep response. Just to give you some example of how venetoclax is working differently from the BCR pathway inhibitors, I think the thing that makes it stand out is the depth of response and the ability to stop treatment. So just to give you one example of this phase 1b study of venetoclax and rituximab combination in relapsed refractory CLL, so you can see that the response rate is excellent. I didn't show that because it was already shown by Dr. Venugopal, but I want to point out that there is a pretty high rate of MRD negative response. So among the CR patients, so you can see that among the CR patients and PR patients, there's a total of 55% of MRD negativity. Why is that important? Because if you're able to achieve a negative MRD status, whether it's PR or CR, you can have a very good duration of response as compared to a patient who had a positive MRD status. So for patients who are able to achieve MRD negative status, this study actually allowed patients to choose either to continue on the venetoclax single agent treatment or actually to stop the treatment. And we have learned from this study that for patients who are stopping the venetoclax treatment after achieving MRD negative response, they seem to be doing fairly well. So this is just an example of showing you a couple of patients who had stopped venetoclax treatment. So the green bar shows you the time they were on the treatment. The blue bar is showing you the time they were off treatment and still in remission. So among these 12 patients, only 2 of them actually relapsed after being off venetoclax for 2 years. And both of those patients were MRD positive patients. So all of the MRD negative patients at the time of report still maintained their response despite being off treatment. So this is making them quite distinct from the BCR pathway inhibitors because with the ibrutinib or the adalimumab treatment, it's very rare to be able to achieve a complete response, and it's extremely rare to be able to achieve an MRD negative response. But with the venetoclax-based treatment, you're able to achieve a very deep response, and therefore this might allow you the opportunity to actually stop treatment after duration of therapy. So we talked about the BCR pathway inhibitors, we talked about the BCL2 inhibitors. There are some other targets that are being studied. I think this one seemed to be very interesting. The ROR1 is a stem cell antigen. So in this early phase 1 study, this molecule is a monoclonal antibody against ROR1 stem cell antigen. And this, by blocking this antigen, you can actually block the stem cell, or what's called stemness signature in the CLL. Just a very interesting concept. So in this phase 1 trial, you can see that this is the response rate in terms of the lymphocyte reduction and in terms of the lymph node reduction. And this is the progression-free survival with a median of 262 days. So moderate activity, but since the idea is that you're targeting the CLL stem cell, it's an interesting concept, and it might be an opportunity to combine with some of the other treatments. Now let's shift gear to talk about immunotherapies in CLL. So like I said, we have used monoclonal antibodies, in particular the anti-CD20 monoclonal antibody for the past 2 decades. And the one we're most familiar with is the rituximab. But there are also 2 other monoclonal anti-CD20 monoclonal antibody that's approved for CLL, the ofatumumab, sorry, it should be in mAb, and the obinutuzumab. And then there's also this recent one, the oblixtumab. Did I say it right? All these different names, right? So the later 2 are more glyco-engineered monoclonal antibody that seem to have improved the Fc affinity Fc receptor affinity, so therefore may improve the ADCC and also some direct cell killing activity. There are also other monoclonal antibodies. For example, there are anti-CD37 monoclonal antibodies. There was also an anti-CD19 monoclonal antibody that was being studied. So very early data. And then now there's also bispecific antibodies that targets both CD19, a B cell antigen, and CD3, a T cell antigen. So this is in a way similar to the blinatumomab in ALL, where you're trying to conjugate, so bring the T cells into the vicinity of the malignant B cells and therefore triggering the cellular immune response. Except that this is not a peptide, this is antibody-based, so it does not require this continuous infusion. So there are clinical trials currently ongoing using the bispecific antibodies that include CLL patients and other lymphoma patients. Checkpoint inhibitors, such as the PD-1 or PD-L1 inhibitors. So this has been is being studied in CLL. There was one study from the Mayo Clinic group that reported a study in relapsed refractory CLL, including Richter transformation. Interestingly, their observation was that it seemed to not have much effect on the CLL per se. However, it seemed to have some effect on the diffuse large B cell component of the Richter transformation. So that, even though it's a very small study, but it does open up a very interesting potential option for Richter transformation, a disease we really don't have any really good treatment for. Immunomodulatory drugs, the IMiDs, so we know that the lenalidomide have been studied in myeloma, in lymphoma. It has also been studied for quite a long time in CLL, and it definitely shows activity. However, I think because of all the other novel targeted agents coming up in the last several years, I think that the development of lenalidomide in CLL has been not as expected. There also the next generation of IMiDs, the CC122, which is a first-in-class pleiotropic pathway inhibitors that seem to have interesting activity. And then there are the CAR T therapy, which I'll spend a bit more time on. And there are also CLL vaccine studies using the patient's own leukemia cells or leukemic-generated antigens, trying to stimulate the anti-leukemic effect. So this is just to show you the lenalidomide data that's giving you so lenalidomide alone versus a combination with rituximab R-squared. The overall response rate improved from the 30% range to the 66% range. We don't have much data on the progression-free survival for that, but the median time to progression-free survival was about 17 months with this particular combination. So next couple slides, we're going to talk about CAR T therapy. So you probably have heard of CAR T from the ALL world and from the diffuse large B-cell lymphoma world, both of which already had FDA approval. However, the first successful CAR T treatment report came from CLL, ironically. Several years ago, Carl June's group reported a single successful case of a highly refractory CLL patient achieving a CR with CAR T treatment. So that was published at New England Journal of Medicine. I think that really started the CAR T era. It caused a lot of attention. However, since then, the CLL CAR T has been kind of lagging behind the other CAR Ts, probably because CLL, we have so many other good treatments available. There's not as much of a unmet medical need compared to the other more aggressive diseases. But now I think we're starting to catch up. So now there are several CAR T CLL studies that are being planned and hopefully will open soon. And we're going to actually open some of those CLL CAR T studies at Northwestern as well. So what is CAR T? CAR T stands for chimeric antigen receptor modified T cell therapy. So this is an adaptive immunotherapy. The idea is that we believe that the T cells is probably the most potent immunotherapy for anti-lymphoma. However, your autologous T cells are not able to recognize or efficiently mount a response. So you can harvest the patient's T cells and engineer and express a chimeric antibody that in the extracellular domain can bind to the target antigen. For example, in the B cells, it would be CD19. So it's like a CD19 antibody on the outside, on the extracellular domain. And the intracellular domain has co-stimulatory domains that can help to activate the T cells. So once the receptor binds to the tumor antigen, this will stimulate the T cells to be activated and to actually expand and to actually have a persistent anti-tumor CAR T in the patient's body that has a constant immune surveillance. So it has shown a very promising response even among the very refractory group of CLL patients. You can see this is the lymph node response. Most of the patients do have a lymph node response by reducing the size of the lymph nodes. And you can see that this is the progression-free survival based on the different response. But this is very interesting. So if a patient either have a CR or PR compared to those patients who had no response, so those patients who had CR or PR seem to have a very good overall survival, of course, despite a short follow-up, compared to those patients who had no response. So even if you had a PR, it seemed to definitely improve your overall survival. So many of those patients, this is about half of the patients were actually able to achieve MRD negative response. So here they're using the PCR to detect IgH. So if it's MRD negative, you can see this is the progression-free survival for MRD negative patients, and this is the progression-free survival for MRD positive patients. So if you're able to achieve MRD negative response with a CAR T, then it seemed to be quite an excellent progression-free survival and overall survival. So the future is really bright. By the way, this is not me. I wish I could do that. It's my daughter. But with all of the available treatments in the pipeline, and I think there are many new things coming, I think the treatment for CLL is really going to be revolutionized in the coming years. And the patients hopefully will have a much better, longer survival. All right, thank you very much.